Confusion, confusion, confusion. You don't like confusion, you avoid. But confusion likes you. You can't avoid. Yes, when we are talking about the chapter from class 11 biology, structural organization in animals, and the topic is animal tissue, it's quite complex and confusing, right? Because the chapter deals with multiple types of tissue and their examples. So, to make it easier to learn it in a proper way with multiple tricks, right? Do not forget to check the questions and DPPs which are given into PW app. Yes, you have to explore our PW app to get some real practice hits. Remember, our learning is not complete if we are not practicing enough number of questions. So, what are the topics we are gonna study in this particular chapter? We'll cover animal tissue. We'll cover different types of animal tissue. Basically, there are 4 types: epithelial tissue, connective tissue, muscular tissue, or muscle, and neural tissue. Right? Okay. So, let's introduce tissue. So, when you talk about the term tissue, what do you mean by tissue? So, you will answer, sir, tissue means group of cell with similar function, similar structure, right, from your previous learning. But you are preparing for NEET, my bro, my sisters, my friends. So, here a perfect definition of tissue. So, obviously, when we are talking about tissue, it should be a group of cells should be there. Okay. But the next criteria, the group of cells, they are connected or they are attached with each other with the help of intercellular matrix. Attached by intercellular matrix. Intercellular matrix. This intercellular matrix acting as biological cement or ground substance to hold these cells together for successful formation of tissue. Yes, these are our cells. So, until unless you are attaching them with the help of cementing material, so if you are building a wall, right, you need some bricks. Bricks looks quite similar, they're of similar origin, but if you are not putting cement in between them, successfully you will not be making a wall, right? So here, if you want to make the tissue, you need biological cement. And this biological cement is also known as intercellular matrix. Okay. What else? The cells should be of same origin. Now you will tell me that, sir, the cell should be of similar shape, size, and function. Okay, let's talk about that. When you talk about that, a group of cells with similar structure and function. Okay. Now, in order to form a tissue, you need a group of cells, but the structural and functional similarity, that is not a mandatory criteria. Remember it. Okay. The similarities in structure and function, that could be that maybe yes or maybe no. For example, there are certain tissue where you will be finding they are of similar origin, they are interconnected by intercellular matrix, okay, they are obviously a group of cells, but their structure and function that is not similar. For example, a tissue where structure and function are not similar, example, blood. Blood is also a tissue, right? Blood is also a fluid connective tissue. But when you talk about the cells of blood, blood has RBC, blood has WBC, blood has platelets, broken piece of cells. So these are different types of cells present into blood, RBC, WBC, platelets. See, the function and structure of RBC is quite different from WBC and platelets, right? RBC is mainly responsible for carrying the gases, oxygen and carbon dioxide, because RBC has hemoglobin molecules inside it. WBC, they are called as soldiers. They are responsible for making the immune system of our body. And platelets, they are responsible for blood clotting when we are getting any sort of wound, right? So their structures also very different from each other, and their function is also different from each other. But still, this group of cells, they are forming a tissue called blood, right? So that's why when you are defining tissue, remember what are the mandatory criteria? A group of cells, they are attached through intercellular matrix, very important criteria. Okay, their origin should be similar, same, but which is not mandatory criteria to define a tissue, that is similar structure or similar function, that may or may not be, right? Okay, now let's move forward. So when we are talking about the formation of tissue inside our body, or if you observe the evolution of different types of animals in animal kingdom, okay, then a very important thing I am telling that all the animals are multicellular in nature. But if I ask you, can you name a group of animal which is multicellular but devoid of tissue, means tissue is absent in which group of animals, right? So the answer is porifera or sponges. Porifera or sponges, for them tissue is absent. We call their level of organization as cell aggregate level of organization, means aggregation of multiple cells with similar origin, few are of similar structure, similar function, but they are not attached, those cells of a porifera not attached by by intercellular matrix or intercellular ground substance. That's why those multiple cells present into the body of sponges, they're not qualified for tissue level, right? Okay, now if you ask me, sir, then which is the group of animals for the first time we can observe tissue into them? Okay, so they are cnidarians or coelenteratans. Cnidaria, this group of animal, for the first time you can observe tissue into them. Like if we talk about cnidaria, the examples are hydra or jellyfish. They are examples of cnidarians. So first animals with tissue level, remember them, right? Okay, now what about us? Let's start it from the time of embryonic development or embryogenesis, right? So it's a fusion of sperm and ovum that will give rise to a zygote, right? And this zygote will give rise to embryo into the mother's womb. So into that mother's womb, gradually the embryo is developing and gradually the tissues will also develop. And we call those tissues as embryonic tissues or embryonic germ layers. So if you observe mother into her womb, a baby is developing gradually. And the developing baby, we call it embryo, right? Now, if we talk about this embryo, it has some uh, initial tissues present into it. Okay. So initially, if you observe a specific stage of embryo, that embryonic stage is called gastrula stage, a specific embryonic stage. The embryonic stage is like that. If you observe their like embryonic development, first it's a zygote, then 8 to 16 cell stage that is called morula. Then beyond 16 cell stage that is called blastocyst or blastula, and the blastocyst, after the implantation with the mother's uterine wall, it will give rise a tri-layered embryonic stage with 3 layers, that is called gastrula. And in this gastrula we'll get some germ layers or initial tissues. The outermost layer, that is called ectoderm. The middle layer, that is called mesoderm, and the inner layer, that is called endoderm. And these are called embryonic germ layers. And by the process of proliferation and differentiation of these embryonic germ layers, 4 types of tissues will be produced into mature human body. So most of the advanced multicellular animals, though all the animals are multicellular, most of the advanced animal, you will find that inside them you will be finding 4 types of tissue: epithelial tissue, connective tissue, muscle tissue, and neural tissue. They are derivative of this either ectoderm, mesoderm, or endoderm. So these initial layers of tissue, they are called embryonic germ layers, right? Again, outermost layer ectoderm, middle layer mesoderm, innermost layer endoderm, right? Now on further developmental process, the ectoderm will give rise few tissue, mesoderm will also give rise few other tissue, other type of tissue, endoderm also will give rise few other type of tissue. Okay. Now if we talk about ectoderm, on further embryonic development, the ectoderm will give rise epithelial tissue, the ectoderm will also give rise neural tissue. Okay. When you talk about mesoderm, the mesoderm can give rise to epithelial tissue. The mesoderm can give rise muscle tissue. Or the mesoderm also can give rise to connective tissue. When you talk about endoderms, endoderm can give rise to again epithelial tissue. Right. So you can observe epithelial tissue can be formed from ectoderm, mesoderm and endoderm. Neural tissue only from ectoderm. Muscular tissue and connective tissue only from mesoderm. Right. Okay. Now let's move forward. So now it's time to make a proper tabulated version of 4 types of animal tissue. Okay. So the first tissue will be formed during the time of embryonic development, that is epithelial tissue. First tissue that is formed, that is epithelial tissue. Basically epithelial tissue that is responsible for making the covering of a body. So thing is that if you are buying a piece of land to build your house, then what you will do, first thing you will be doing, that is to make a fence or to make a boundary wall to protect your land, right? So when our body is getting developed, so the first priority is to protect our body from external environment. That's why first epithelial tissue is produced. Okay. Now once epithelium or epithelial tissue is formed, once this epithelial tissue is formed, the second tissue that is produced, that is connective tissue. Now why is it's connective tissue? Now once you form the epithelium, now second thing you need to link different types of tissue. During the time of embryonic development, the organs and systems and the tissues are differentiating, forming different types of organs, systems and tissues, right? So you need to supply nutrient to them, you need to supply oxygen to them, you need to eliminate metabolic waste from them. So in order to link them, link different types of tissue, you need a sort of tissue which has this capability, that is connective tissue. Okay. Third tissue that is formed, that is muscular tissue. Why muscular tissue is important? Because the muscles will give us the capability of movement and locomotion. So body will get mobility, okay, due to the presence of muscular tissue. And the very last tissue that is formed, that is neural tissue. The neural tissue is responsible for control and coordination activities. It's responsible for sensitivity. You need to sense the internal environment or external environment, right? So that's why these are the 4 basic types of tissue present in our body. If we talk about different organs, different system, but the basis is these 4 types of tissue. Okay. So as per our discussion, what we are saying, this epithelial tissue, this is the first tissue to formed, first tissue to be formed during the time of embryonic development, right? Epithelial tissue is responsible for making the covering of our body, maybe the outer lining or maybe the inner lining of our body and organs. So basically they're responsible for making the covering. Okay, and epithelial tissue, if we talk about their origin, their ectomesoendodermal in origin. Remember the embryonic germ layers, ectoderm, mesoderm, endoderm. All these three types of embryonic germ layer can give rise to different types of epithelial tissue. So that's why origin is ectomesoendodermal. So ectomesoendodermal, that is their origin. Right. Now let's come to the connective tissue. They're responsible for linking and supporting. Okay. Their origin of connective tissue, mostly they are mesodermal in origin. Mesodermal in origin, connective tissue. See, now this epithelial tissue and connective tissue, they have good power of regeneration. The highest power of regeneration is seen in epithelium or epithelial tissue. Even the tissue of connective tissue, they also have very good power of regeneration. Okay. Regeneration means you have watched the movie of X-Men, Wolverine, right? So whenever he is getting any scratch or wound, within few seconds, I think, within few seconds, the Wolverine is able to recover the wound or scratches. Similarly, if we are also getting scratches or wound, it takes few days to get recovered, right? So because basically our process of regeneration is very slow, but the process of regeneration was very fast in Wolverine. So sometime there will be damage in our muscles, damage in our bones, damage in our skin, but gradually they will recover due to this regeneration capacity. Muscular tissue, they are responsible for locomotion and movement. And if we talk about the origin of muscular tissue, then the origin is again mesodermal. And for neural tissue is responsible for sensitivity, for control and coordination. The origin of neural tissue is ectodermal. Remember it. Okay. Now we'll learn about detail. First we'll learn epithelial tissue. We'll learn in detail about epithelial tissue, connective tissue, muscular tissue and neural tissue. Okay. So first, epithelial tissue. Now, whenever you are studying biology and you are getting a new terminology, you just have to split up the term and you have to search for the Greek or Latin origin of that particular name. So when you talk about epithelial tissue, right? Epithelial tissue, epi means upon and thelia means to grow or to cover. So basically, epithelial tissue always cover or forms the inner and outer lining of our body. That's why we are naming it epithelial tissue, and always they will grow upon connective tissue, right? Okay, so basic features of epithelial tissue. They forms inner or outer boundary lining of body or organs, right? Next, epithelial tissue, they have highest regeneration capacity. They're the first tissue to develop during the time of our body development. Okay. They are avascular tissue. They're avascular tissue is because they are avascular. Avascular means there is very limited or no blood supply present. Okay. Now, why they are avascular? Because they are making the boundary lining of our body. The cells are very tightly packed or compact. That's why more tight packing, less amount of intercellular matrix will be there. So they have very less amount of intercellular matrix present into them, as because they are very tightly packed. The cells are very tightly packed. Tightly packed cells are very compact, as because they are making the boundary lining. So that's why very rarely blood vessel will enter into them. So as because they don't have blood vessel, we are calling them avascular. So that's why grow upon connective tissue. Always they are growing upon connective tissue, and there is a basement membrane which links the connective tissue and the epithelial tissue. That basement membrane links connective tissue to epithelium to supply nutrients and all other components, right? So these are the very basic features of epithelial tissue. Now, if we talk about their types, the types of epithelial tissue, they're mainly of two types: simple epithelium and compound epithelium. Now, we'll study in detail about simple epithelium and compound epithelium. Different diagrams of simple epithelium is given here. Basically, when the epithelial cells are arranged in a single line, that is called simple epithelium. When there will be multiple layers of cell present into the epithelial tissue, then we call that tissue as compound epithelium, right? So when You will observe flattened cells. We name this epithelium as squamous epithelium. When you observe cube-like cells, we name it cuboidal epithelium. When you observe pillar-like, elongated, slender cells, we name it columnar epithelium. Right. So these are different basic kind of cells what you are gonna get inside the epithelial tissue. Squamous cells, flat cells, cube-shaped cells or cuboidal cells, elongated, slender cells called columnar cells. Right. Okay. Now before this I was telling that these cells of epithelium, they are avascular, that's why always they grow upon connective tissue. Suppose these are epithelial cells. So they will rest upon connective tissue layer. And basement membrane will link them up. Right. Okay. So these are epithelial cells. This is connective tissue. This is basement layer or basement membrane. And this connective tissue that has well supply of blood vessels or blood capillaries. Right. So through basement membrane, nutrients and other essential things are getting diffused towards the epithelial cells. Okay. Now, the free surface, this is called apical region, apical surface or apex. This surface, this is called basal surface. So these epithelial cells from their basal surface, they are resting upon connective tissue and the apical surface is free. Okay, that's why we can say that the free surface either face environment or atmosphere or air or water or it may face body fluids. Basically, when the free surface is present outside, outwards, like our skin that is facing this environment, the air. For a fish, the outer surface is facing the water. Okay, but blood vessel is also made by the inner lining of blood vessels is made by epithelial tissue. So those endothelial cells, the inner lining of blood vessels that is facing blood or body fluid. So when they are facing body fluid, that basically they are present in inner surface. So always remember that epithelial, the cells of epithelial tissue always has a free surface, always has a free surface, right? And that free surface, either it may face the environment or it may face the body fluid. If the surface is outside, it will face the environment, either air or water. If the surface is inside, then that will face body fluid. Okay. So basically, first we'll talk about simple epithelium. Simple epithelium means epithelial cells with a single line. So that is called simple epithelium. One layer of epithelial cells. That is called simple epithelium. Okay. Now simple epithelium, there are three types. Simple epithelium, there are three types. Simple squamous epithelium, simple cuboidal epithelium, simple columnar epithelium, and there is also another subtype. It is basically modification of simple columnar. You can call it pseudo-stratified. Okay. When you talk about simple squamous epithelium, the cells are of they're like tiles, but with irregular boundary, flat cells. Flat cells with irregular boundary. See the boundary, there is no regularity into them. Flat cells with irregular boundary. This is the nature of cells present in the simple squamous epithelium. That's why from the scale or from the flattened shape structure, we got the name simple squamous. Okay, and their main function, they help in diffusion. They will be forming diffusion barrier. Is their main function. They are responsible for formation of diffusion barrier. Okay. Now I'm coming to the example with a trick. We'll explain later on. Remember the term beta. Okay, now I'm coming to simple cuboidal epithelium. Simple cuboidal epithelium. Cells are of cube shape. Right, and they're responsible for mainly secretion and absorption is their main function. Secretion and absorption. Basically, they help to form a glandular part or absorptive part, right? Okay. And one more important thing about these simple cuboidal cells, you will find them into the germinal epithelium of testis and ovary. Basically, they're responsible for forming sperm and ovum, male gametes and female gametes. So that's why simple cuboidal epithelial cells are also called as germinal epithelium. Germinal epithelium. Why? Because they're responsible for formation of the germinal cells in testes and ovaries, which will give rise to sperms and ovum, right? Okay. Their trick, again, I'm gonna explain in further slides. For simple cuboidal epithelial cells, quota, beta is going to quota, right? Now let's come to the simple columnar epithelium. Now thereof cells of this like this bottle, elongated shape. This is the apical surface, this is the basal surface. And the nucleus will be present towards the base for the cells present into the simple columnar epithelium. Okay. Elongated cells, you can say this cells is of pillar like cells, elongated with basal nucleus, means nucleus present the base, at the base, this is the apex. Towards the base you will find the nucleus. And about their function, when you talk about their function, that is similar to the function of simple cuboidal epithelium. Simple cuboidal cells, they are also responsible for secretion and absorption. Cells of simple columnar epithelium, they are also responsible for secretion and absorption. So that is the common function of simple cuboidal and simple columnar epithelium. Okay. Now, if I talk about the example of simple columnar epithelium, beta has gone to quota. Now, after going quota, whatever like he will study from his sir, and also he will get a GF, girlfriend. That is the trick to remember all the examples of simple epithelial tissue. Okay. Now, what is pseudo stratified? We'll come into that. First, we'll discuss about the examples of simple squamous, simple cuboidal, and simple columnar, as because they are given in NCERT. The pseudo stratified because as they are kind of modified columnar, right? A kind of modified columnar tissue. They're not explained in NCERT. And pseudostratified tissue you will be find into the inner lining of trachea, into the pharynx. So in trachea and pharynx, you will find this kind of a special kind of modified columnar epithelial tissue with some cilia and all. We call it pseudo stratified. Sometime it got a nickname, pseudo stratified ciliated columnar glandular epithelium, PSCCGE, pseudo stratified ciliated columnar glandular epithelium, right? Okay, so what I was telling, I was talking about the example of simple squamous. Remember the trick? Beta. B for Bowman's capsule. There is a part of nephron, this cup-like structure. So this cup-like structure of nephron, that is made by flattened cells, and we call it Bowman's capsule. Okay. And what is the abbreviation of this E? E means endothelium. What is endothelium? Inner lining of blood vessels. Endothelium means inner lining of blood vessels, or even sometime the endocardium, the inner lining of heart. There is also kind of endothelium only. Okay. T for thin segment of loop of Henle. Thin segment of loop of Henle, this part. Loop of Henle also a part of nephron. So majority of the tissue of nephron, which is a structural and functional unit of kidney, that is made by a columnar epithelium. But when you talk about the Bowman's capsule and the thin part of loop of Henle, the hairpin or U-shaped loop, it has thick part as well as thin part. The thin part, that is also made by simple squamous epithelium. Okay. Okay. Now let's come to the A. A means alveoli or air sacs of lung, right? So when you talk about lungs, inside our two lungs, we have near about 300 millions of air sacs. Inside our lungs, each lung has 150 millions of grape bunch or bunch of balloons like air sacs. we call them alveoli. And if you enlarge the view, it will be like this. This is one alveoli, and the alveoli has a few squamous cells, and this is surrounded by alveolar capillaries. So, what I said, what is the function of simple squamous epithelium? Formation of diffusion boundary. So, diffusion of oxygen and carbon dioxide will take place through these layers of squamous epithelial tissue, right? In between the alveolar air and blood of alveolar capillaries. So, the function is clear for simple squamous epithelium. It helps in the process of diffusion. Even here also, when the filtrate for urine formation is forming inside the Bowman's capsule, you need diffusion of solute and solvents in between the glomerular capillaries and the epithelial by crossing the epithelial layers of this Bowman's capsule, right? So, this is the abbreviation of the term beta. Now, beta has gone to where? Quota. Now, with quota, we'll get the examples of simple cuboidal epithelium. Okay, one more thing I'll be adding here. Beta, after going quota, he has changed the DP of her Insta profile. Okay, right? So, K for kidney tubules, the pipeline or the tubes or tiny tubes present into the kidney, that is a major part of nephron, we call them kidney tubules. And we talk about kidney tubules, we must talk about DCT, distal convoluted tubule, and PCT, proximal convoluted tubule. Okay. Now, O for ovum, ovary, T for testis, right? Now we know that ovary and testis, they have germinal epithelium. They have germinal cells to produce gametes. So that's why ovary and testis, they also have simple cuboidal epithelium. A for acini of pancreas. This is a special tissue which are responsible for formation of pancreatic juice. So acini of pancreas, they are responsible for production of pancreatic juice, which help us in our digestion process, right? So these are examples of simple cuboidal epithelium. Okay, from these letter D, you can also get the ducts of glands, like small ducts of salivary glands, sweat glands, their pancreatic duct, they're also made by simple cuboidal epithelium. Okay. Now, what about the examples of simple columnar? Epithelium. Remember the trick? Sir GF, after going quota, the boy changed the DP and he went to study from his sir, and he also got a girlfriend. Sir, stomach, inner lining of stomach, right? So stomach has the gastric glands, that is called inner lining of stomach, that also made up of simple columnar epithelium. I for inner lining of intestine, inner lining of intestine. R for respiratory tract. G for goblet cells. And F for fallopian tube. All these structures in the inner lining, you are going to get simple columnar epithelium. Okay. Now, especially if we talk about the respiratory tract and fallopian tube, then there will be modification of simple columnar epithelium. The simple columnar cells will be with cilia, and we call it ciliated columnar epithelium. In respiratory tract, we'll get ciliated columnar epithelium. Right? Similarly, in fallopian tube also, you will get the similar kind of cell. This ciliary movement in respiratory tract help in the transportation of air and mucus, and the ciliary movement of the cells of fallopian tube that helps into the transportation of the ovum towards the zone of fertilization. Okay. Now, if I'm talking about goblet cell, goblet means the wine glass. The cells, they look like wine glass, and they produce mucus. So goblet cells, this is a goblet cell, look like wine glass. They're responsible for production of mucus. They're also present into our respiratory and digestive tract. Okay. So this was all about simple columnar epithelium. Now some modification can be observed for simple cuboidal and simple columnar, right? If a cell has the cilia, then we will call it ciliated cell. It can be ciliated columnar or ciliated cuboidal. If the cell has microvilli, the finger-like projection, we call it brush border. Either it can be brush border cuboidal or it can be brush border columnar. Okay. So if someone asks you, give some example of ciliated epithelium with cube-like cells, then the examples will be neck of collecting duct, there is also a structure of neuron. Ciliated epithelium with columnar cells, with elongated cells, then obviously respiratory tract. according to NCERT, and fallopian tube. If someone asks you brush border cuboidal epithelium example, then you can answer proximal convoluted tubule, PCT of kidney, PCT of nephron of kidney. Examples of brush border columnar epithelium, then inner lining of stomach, and intestine. You know that there are villi and microvilli present on the inner lining of small intestine where absorption of food is taking place. So those cells are called enterocytes. They are this brush border columnar epithelial cells, right? Okay. Now in some cells you are gonna get some cilia, but this cone kind of ciliated structure, but they are non-motile cilia. We call these cilia as stereocilia. So there are few cells with stereocilia. Basically, what is the function of this particular cilia? This particular cilia, we call them kinocilia. They help in the movement of particles, right? Microvilli, they increase the surface area wherever there will be absorption, right? PCT inside the nephron, the highest reabsorption takes place. Wherever there will be absorption or reabsorption taking place, you need to increase the surface area. So you can support the structure or support the surface with microvilli, either microvilli or stereocilia, right? Okay. Stereocilia, remember, is immovable cilia. Kinocilia, they are movable cilia. Stereocilia, they are kind of microvilli because there is a similarity between stereocilia and microvilli. They both are extension of their cytoplasm or protoplasm. Okay. Now, the tissue pseudo-stratified ciliated columnar glandular epithelium. Though it's not given in NCERT, we can omit this part. Basically, what happens in pseudo-stratified tissue, stratification means layer by layer arrangement. So these have some elongated columnar cells. This tissue has some elongated columnar cells, and in between them some smaller cells are present. So that's why the location of their nucleus will differ. So when you are observing under microscope, due to the location of different location of nucleus and their different shape, what you will observe, you will think that there are two layers of cells. You will think that this is arrangement of two layers of cell, but basically there is a mistake of our eye, right? Basically there is a single layer of cell. Few cells are elongated and longer, and few cells are shorter, right? So that's why we are calling it pseudo means false. Layer is observed, but actually in real there is a single layer, but in observation under microscope you will observe two layers. So that's why you are calling it pseudo stratified. Okay, as because cilia is present on the apical surface of this columnar cells, so ciliated, and some goblet cells are also present in between them. So that's why glandular epithelium. You will find them into the inner wall of trachea and pharynx. Okay, and to some extent into the inner wall of bronchi. Now, the next thing we are going to study, that is glandular epithelium. Are there different type of tissue? No. Basically, when you are talking about glandular epithelium, they are modified cuboidal or columnar epithelium. What do you mean by gland? Gland means expert or specialized in secretion. So glands are specialized for secretion. So few cuboidal and columnar epithelial tissue will undergo modification and they will form glands, right? So that's why we are calling them glandular epithelium. Now let's discuss their types. See, in different angle you can classify glands. Either the glands can be unicellular gland or the glands can be of multicellular gland. See, just now we were talking about goblet cells. So this is a goblet cell, a glass. They look like glass, wine glass. So wine glass shaped cells are called goblet cells. They are examples of, this is the nucleus. These are examples of unicellular gland. Goblet, sorry, not goblet, goblet cell. Yes, responsible for production of mucus. This is the mucus secreted by them. Goblet cell. Where they are present? They are present in the inner lining of our gut, means alimentary canal, and also inner lining of our respiratory passage. So we are talking about glandular epithelium, and glands are of two types, unicellular glands and multicellular glands. And one of the example or prominent example of unicellular gland, that is goblet cell. Yes, goblet cells. This is the histological diagram of goblet cells, which looks like the wine glass. Now, another example of unicellular gland, they are Paneth cell. Okay. Now, these goblet cells are basically responsible for secretion of mucus. Goblet cells, they are responsible for secretion of mucus, and Paneth cells, they are present in the inner lining of our intestinal wall, and this Paneth cell, they are responsible for secretion of lysozyme. They have antimicrobial property, right? Okay. Now, apart from these two glands or these two unicellular glands, all other glands which you were knowing about, they belong to this multicellular group, right? So when I'm telling multicellular glands, all the glands you can imagine, they fall under this particular category. So some examples like pituitary gland, multicellular gland, sweat gland, multicellular gland, sebaceous gland, multicellular gland. Cerumen gland, multicellular gland. Cerumen gland, which is responsible for secretion of ear wax. Sweat gland, responsible for secretion of sweat. Pituitary, this is the master gland of our body, one of the most prominent endocrine gland. Sebaceous gland, which is responsible for secretion of sebum or oil. Okay. Gastric gland. Pancreatic acini, all are responsible for secretion of gastric juices. Like gastric gland will secrete hydrochloric acid, pepsinogen, right? Okay. Gastric lipase, pancreatic acini, they are responsible for secretion of pancreatic juice. So all other glands you can imagine in our body, apart from goblet cells and Paneth cells, they are goblet cells and Paneth cells, they are unicellular gland, and apart from these two, all are multicellular glands. Okay. So here one of the example of multicellular gland is given. The criteria to form multicellular gland, that is made by numerous cells like gastric glands present into our stomach. So this is example of multicellular gland, and this is example of unicellular gland. Okay. Now we'll do further classification of glandular epithelium, which are modified columnar or cuboidal epithelium, right? So glands can also be classified basis on their secretion or pouring of their products. Glands are specialized for secretion, right? So based upon the type of secretion, and pouring, which we call it crine activity. The Latin term we use against this, that is crine. Glands are of two types, mainly endocrine gland and exocrine gland. And there is another third type of glands are there, which are mixture of endocrine and exocrine. We call them heterocrine gland or dual gland. Okay. Now let's discuss about them: endocrine, exocrine, heterocrine. Okay. When you are talking about endocrine, these are the ductless gland. They secrete their product secretion inside the gland only. Okay. So if there is a gland, say this is the gland, this is the gland. The cells of this gland are endocrine cells. are secreting something, they will be secreting the product inside the gland itself, and they will release the product with the help of blood stream to the targeted tissue. They will release the product into the bloodstream and send it to the target, right? So endocrine glands are ductless glands without ducts. They don't have pipeline to secrete their product. That's why they're secreting inside the gland only. Okay, their product is hormones or the chemical messenger. The product of endocrine glands are hormones, or we can call them hormone releasing gland. So all the hormone releasing glands will come under this category of endocrine gland. Like for example, starting for our hypothalamus, pituitary, thyroid, pineal, parathyroid, adrenal. These glands produces or releases secretes only hormones. That's why these are pure endocrine glands. Okay. Now let's talk about exocrine gland. Now when I'm talking about exocrine gland, these glands have duct system to release their product. Suppose this is the gland. The product will be released by duct. The product will be released by duct. So these are glands with duct, release the product, secrete the product with the help of pipeline or duct. Okay. The secretion, when I'm talking about their secretion, when I'm talking about the secretion of this particular gland, they secrete enzymes, digestive, hydrochloric acid, oil, milk, sweat, ear wax. So all these product are secreted by exocrine gland. Now if I talk about enzyme, they secreted by digestive glands, like gastric gland secretes pepsinogen or pepsin. HCl also secreted by gastric gland present into the stomach. Oil secreted by sebaceous gland. Milk secreted by mammary gland. They're basically mammary gland modified sweat gland. Sweat secreted by sweat gland, and earwax secreted by cerumen gland or ceruminous gland present into our external ear. Okay, so all these glands are exocrine gland. Okay, if I'm talking about those unicellular glands, remember goblet cells and Paneth cells, they also will fall under this exocrine part. So secretion of mucus is also with the help of goblet cells are under exocrine glands, right? Now heterocrine glands. Now focus your mindset. We are talking about heterocrine gland. Heterocrine means they have endocrine secretion as well as exocrine secretion. So that's why I'm calling them a heterocrine or mixocrine or dual gland. And remember the trick OTP. Examples of heterocrine gland. Okay. O for ovary, T for testis, and P for pancreas. These are some famous heterocrine glands, right? Ovary secretes some hormones as well as it secretes the secondary oocyte. Testes secrete sperms as exocrine activity, secretion of testosterone as endocrine activity. Pancreas, yes, pancreas, it has two kinds of secretion. Exocrine part is called pancreatic acini and endocrine part is called islets of Langerhans or pancreatic islets, right? Acini, they're the exocrine part. Islets, they're the endocrine part. This islet secretes hormones like insulin, glucagon, somatostatin, pancreatic polypeptide, etc. And this pancreatic acini secrete pancreatic alkaline pancreatic juice, right? Okay. So let's move forward to the next kind of tissue. The another type of epithelial tissue, that is compound epithelium. Compound epithelium. Okay. Now let's recap simple epithelium according to NCERT. There are three types of simple epithelium based upon the size and shape of cells. They were squamous epithelium or simple squamous, simple cuboidal, and simple columnar. Okay. Now let's talk about compound epithelium. Here you can see multiple layers of cells. Simple epithelium, it had only one layer of epithelial cells, but if multiple layers of different types of epithelial cells are present into the compound epithelium. Okay. Their main function, the function of compound epithelium is protection. So it's a protective tissue. Compound epithelium is a protective tissue. It protects against different types of physical, chemical, mechanical stress or stresses. Okay. It has limited function in secretion and absorption. Yes, I'm talking about our skin, right? Skin is a dry surface which also comes under a compound epithelium. See, the skin is the protective, acting as a protective organ of our body. It has like less important function in case of secretion and absorption, because secretion and absorption, they are function of simple epithelium, specifically specifically columnar and cuboidal epithelium. But compound epithelium, they are mainly protective tissue. They have uh, they protect us against physical, chemical, mechanical stresses. Okay. They have less known function in secretion and absorption. Secretion and absorption, again and again I am telling, these two are the function of simple epithelium and specifically columnar and cuboidal epithelium. Yes. Now we'll talk about the types of compound epithelium. So compound epithelium there of two types: transitional and non-transitional. Non-transitional is also called stratified epithelium. Right? Transitional and non-transitional or stratified. Okay. Now, when I'm talking about transitional epithelium, the shape of this particular, the cells of particular tissue transitional epithelium, the shape shows transition from sometime they you will observe them, they are looking like cuboidal cells, but upon stretch or upon discharge they will look like columnar. Sometime you will observe them, they are looking like squamous epithelium. So when the cells of tissue, they are performing shape change upon change into stretch, when you are applying force into them, right? When you are applying stretch, when you are stretching this tissue, the cells will undergo their shape change. So this process is called transition, and this transitional epithelium you will find them into your urinary bladder, the wall of urinary bladder, right? You'll find in the wall of renal pelvis. You will find in the wall of ureters. So all are associated with urinary system. So that's why transitional epithelium is also called as urothelium. Transitional epithelium is also called as urothelium. Okay. So when I'm talking about urinary bladder, right? What is the function of urinary bladder? Acting as a storage of urine. So more the urine is getting stored inside this urinary bladder, it will exert force or pressure to the wall of it. Now it must have the capability to stretch the wall of urinary bladder, and due to the exaggerated force which are applied when it's storing the urine inside, the cells present into the inner wall, they will undergo through a transition or shape change. That's why we are calling it transitional epithelium or urothelium. Now let's talk about non-transitional or stratified epithelium. Now what is the meaning of stratification? Stratified means layer by layer arrangement. Layer wise arrangement is called stratification. Like when you are eating a burger, when you are eating the sandwich, you will find layer by layer arrangement. So that is called as stratification. Okay, so stratified layer wise arranged epithelial cells which will not undergo transition, we will call them non-transitional or stratified epithelium. They can again be of compound stratified columnar epithelium, stratified cuboidal epithelium, stratified squamous epithelium. But we need to follow the NCERT. So NCERT is telling about two kind of stratified squamous epithelium tissue, that is skin, and another is the buccal cavity or oral cavity, right? Now when I'm talking about skin, that is example of dry surface. When I'm talking about buccal cavity, that is example of moist surface. So if you are asked, give one example of stratified epithelium or compound epithelium with moist surface, you need to talk about the oral cavity, the lining of oral cavity, And if someone asks you that about the nails, about the hair which are present on skin or the skin or the horns in some animals, they are examples of dry surface or dry stratified epithelium. Okay. Now, why skin is dry but buccal cavity is moist? Okay. The reason is that skin has accumulation of keratin, right? Now, due to the accumulation of keratin, which are responsible for formation of our nails, hairs, and horns in animals. So that's why we call it keratinized epithelium. Skin is also a keratinized stratified epithelium, and buccal cavity there is no presence of keratin, that's why it's also can be called as non-keratinized. Then we are completed with simple epithelium as well as compound epithelium, means epithelial tissue. Now we'll move forward to some questions, right? And one more thing you need to remember about epithelium tissue, we'll learn into the topic of cellular junctions, that epithelial tissue, as because they are forming either the outer boundary or the inner lining of body and organs, as because their cells are very tightly packed, all kind of cellular junctions like tight junctions, adhering junctions, and gap junctions are present into epithelial tissue. Remember, there are majorly three types of cellular junctions. There are certain structure which helps the cells for their closely packedness, right? They're helping the cells to remain closely attached, and those structures are called cell junctions, right? There are majorly of three types: tight junction, adhering junction, and gap junction. So all these three are present. You will be finding them into epithelium or epithelial tissue, right? So let's solve this question. So question is asking, pick the correct option with regards the given below figures. Okay. So by observing the figure, you can correlate with our learnings. This is the number A. This is the diagram of squamous epithelium, flattened cells. These are slender, elongated cells, columnar epithelium. B, option C, cube-like cells, right? Cuboidal epithelium. Option D, these are also columnar cells with cilia, right? So according to our understanding, we'll try to find the correct answer. Okay, so A is squamous epithelium, and its function, we need to find out the correct option. Its function is absorption. No, never. Squamous epithelium is mainly responsible for making the diffusion boundary. You will find them into the alveoli of lungs, remember. Okay, B is cuboidal epithelium. B is not cuboidal epithelium, so it's a wrong one. Option C, columnar epithelium. Option C, columnar epithelium. That's also a wrong answer. Option D, it's a ciliated columnar epithelium. Perfect. It's found in the fallopian tubes. Yes, the ciliary movement of the inner lining of fallopian tube if this is a part of a female reproductive system, the ciliary movement of the inner lining of fallopian tube will help the progression of the secondary oocyte towards the ampulla where the sperms are coming. So if you want a successful fertilization into ampulla region, you need this help of ciliary movement present in the inner wall of this fallopian tube, which is associated structure. Which is one of the important associated structure of female reproductive system. Okay. Now let's move to the next question, a very important question, mesothelium. Okay. Now I was talking about different kinds of epithelium or simple squamous epithelium. So mesothelium is also basically one of the example of simple squamous epithelium. Remember the example of simple squamous epithelium. Okay. Now where we are gonna find this mesothelium? They're the lining of xylem which originated from mesoderm, lining of xylem which originated from ectoderm, lining of heart which is originated from endoderm, or lining of heart which is originated from mesoderm. So first eliminate these answers. They are not the lining of heart. Lining of heart is called endocardium, right? The inner lining of heart is called endocardium. Outer lining is called pericardium. Okay, and all the lining of heart, if I am talking about, is the outer covering pericardium. Then we have epicardium, myocardium, and endocardium. So mesothelium is not related. That mesothelium is obviously the lining of our coelomic cavity or body cavity, right? Okay. So is it originated from mesoderm or ectoderm? So the answer is originated from mesoderm. So this is our correct answer. Okay. Now another question: stretchable or waterproof epithelium. Okay. So when it's stretchable, the answer will be transitional epithelium, a special type of compound epithelium, right? Yes. Another question: the type of epithelial cells which line the inner surface of fallopian tubes, bronchioles, and small bronchi. Okay, lining of fallopian tube, bronchioles, and bronchi. You can guess it, right? Fallopian tube needs ciliary movement. Bronchioles, when we are inhaling or exhaling, we need some help of cilia. So the answer will be ciliated epithelium, the type of epithelial cells which line the inner surface of fallopian tubes, bronchioles, and small bronchi are known as ciliated epithelium. Okay, right? Let's move to the next slide. Another question, and it's saying that goblet cells of alimentary canal are modified form. Yes, we know the answer. Goblet cells, they are wine glass like cells, tall, slender cells, right? They're tall, slender cells responsible for secretion of mucus, right? So these are cells, they are modified columnar epithelium. Goblet cells are modified columnar epithelium. Okay, now we'll learn about next tissue, there is connective tissue. Okay, the most important thing about connective tissue, remember, there is the most abundant and widely spread tissue inside our body, responsible for supporting, giving our body framework, and making the link in between different tissues, right? Without the connective tissue, without the help of connective tissue, successfully we cannot make a full body of an organism, right? So this is the most important issue, whether you are talking about your blood, or you are talking about your lymph, or you are talking about your body fat, or you are talking about your bones, or you are talking about your cartilages. Everything falls under this particular tissue, that is connective tissue. So let's elaborate connective tissue. So when I'm talking about connective tissue, they're the most abundant and widespread tissue during the time of our embryonic development. Just after the formation of epithelial tissue, connective tissue will be formed, right? So they're responsible for providing support, responsible for linking, they are forming the framework of our body, they're responsible for gaseous exchange, delivering the nutrients. So they have multiple works to do. Okay. Now, if you are asked that this connective tissue, are they do they have regeneration capacity? Yes. Highest regeneration capacity is there for epithelial tissue, but the connective tissue, it also posing a good efficiency to regenerate the cells. So it's having the regeneration capacity. Okay. Now, what next? Now we'll talk about the important components of connective tissue. If you want to form a connective tissue, okay. Now, if in your home, if you are forming payasam, right? Or in Hindi we say that kheer, right? If you are forming that, so what are the components you will be needing? So first we will take a bowl. Now inside the bowl we will pour milk, right? Now inside the milk we will put some dry fruits and sevai. Also you can put some rice into that and sugar and all, and we'll be putting some sevai. Now if you assume the medium of that payasam or that kheer, which is making the medium or holding all the things, that is milk, right? Now the milk is containing some dry fruits. Assume that they are the cells of a particular tissue. Now if you consider the fibers of the sevai or the sev, so they are acting as a fiber. So in our body, if we talk about connective tissue also has three components. And those three important components are cells of connective tissue, fibers of connective tissue, and ultimately holding media of cells and fibers, that is ground substance or intercellular matrix So here you can observe a diagram from NCERT is given, which is explaining different components of connective tissue. Here you can find a cell, a very important cell of connective tissue, that is fibroblast cell, right? Here you can find the fibers of connective tissue. Different types of fibers are present if you look inside the connective tissue. And here you can find the intercellular matrix or ground substance, which is holding all the components, right? So let's explore the types of cell, the types of fibers, and the ground substance which will get inside connective tissue. Now if I'm talking about the cells present in connective tissue, you will find fibroblast cell. They're the most important cells of connective tissue. Okay, you are gonna get macrophages, phagocytic cell. You are gonna get neutrophils. In connective tissue, you are gonna get plasma cells. They're responsible for making the antibodies. They're basically modified lymphocytic cells or B cells. Okay, in connective tissue, you will find fat cells or adipocytes. In connective tissue, you are gonna get mast cells. These mast cells, they're responsible for secretion of very important components like heparin, histamine, and serotonin, right? So they're the major cells you're gonna get inside the connective tissue. Fibroblast cell, the cells responsible for secretion of the fibers and the matrix. Phagocytic cells like macrophages and neutrophils. Macrophages and neutrophils. You're gonna get cells like also another kind of WBC, mast cell or modified WBC. They're like basophils of WBC. You're gonna get modified B cells, which are responsible for formation of antibodies, plasma cells, fat forming and fat storing cells, adipocyte cells, right? Okay. Now fibers, three kinds of fiber you're gonna get inside connective tissue. Okay. White fiber, that is called collagen fiber. Yellow fiber or called elastic fiber. And slightly blackish in color or bluish in color or deep red in color, another kind of fiber which looks like a network, that is called reticular fiber. Right, so these three types of fibers we're gonna get inside connective tissue: collagen fiber, elastic fiber, reticular fiber. Okay. Now, if I'm talking about ground substance, the main medium to hold all the cells and fibers produced by fibroblast cell, basically they are made up of hyaluronic acid, and other mucopolysaccharides. Mucopolysaccharides, right? So these are the important component present into the ground substance. The main component is hyaluronic acid and also few other mucopolysaccharides, right? Okay. Now let's elaborate different functions of the cells. So if I'm talking about fibroblast cells, they're responsible for formation of fibers and matrix or ground substance, right? Okay. Now if I'm talking about phagocytic cells, if I'm talking about phagocytic cells, they are macrophages and neutrophils. They will kill the pathogen or stop the pathogenic attack by the process of phagocytosis. They move like amoeba. So these are also can be called as amoeboid cell, right? Okay, we have plasma cells. Plasma cells, they're responsible for formation of antibodies. They're responsible for formation of antibody or antibody forming cells. They are basically modified or matured effector B cells. You will get other lymphocytes, T cell and other B cells. Okay, you will get adipocyte. Adipocytes, they are also called as fat cells, responsible for production of fat and storage of fat. And lastly, we're going to talk about mast cells. must. So their must cell, they behave like basophil. They have similarities with which cell? They have similarities with basophil cells. They're not basophils. They have similarities with basophils. You have heard this song, Baby ko bass pasand hai, and after listening the bass of the song, the baby is feeling must. So basophils are similar to mast cell, and they will secrete the similar components or chemical components released by basophils. So mast cell, they will secrete heparin. Heparin is acting as anti-clotting factor or anticoagulant. That's why inside your body, your blood is not getting clotted. It will secrete serotonin. Serotonin is responsible for vasoconstriction. It's a vasoconstrictor. And histamine is a vasodilator and bronchoconstrictor. So all these things, serotonin and histamine, they are responsible for inflammatory actions of our body. Right. Okay. So we were talking about the cells present into the connective tissue. Now we'll talk about the fibers present into the connective tissue. So we know there are three types of fibers are there mainly. Collagen fiber, they are made up of collagen protein. Elastic fiber, they are made up of elastin protein. And the third type of fiber is reticular fiber. They are made up of reticulin protein, a kind of modified collagen. Okay. So when I'm talking about collagen fiber, they are unbranched fiber. When I'm talking about elastic fiber, they are yellowish branched fiber. When I'm talking about reticular fiber, they are highly branched fiber. That network-like appearance given them the name reticular. Reticule means network. Okay, so collagen, we can see that is inelastic in nature, like a rubber band. You cannot stretch it, but elastic fiber by the name, it's a elastic one. Good elasticity is present into this fiber. Elasticity is present into yellow fiber. This is also called as yellow fiber. Collagen is also called as white fiber. Okay. Now reticular fiber, this will form a network like structure. That's why it has got the name reticular. Okay, it's made of by the protein of reticulin. These are modified collagen only. Right. Okay. Now let's move to the next thing. Now we'll classify different types of connective tissue as because they're the most abundant and widely distributed tissue, right? So we need to classify them. Okay. So connective tissue are divided into two types, connective tissue proper and specialized. But if we follow the NCERT, connective tissue is divided into three types: loose connective tissue, dense connective tissue, and specialized connective tissue. And specialized connective tissue. Okay. Now this loose and dense connective tissue, these are also called as connective tissue proper because this loose and dense connective tissue, they have all the components present which we need to, which you were just learning that connective tissue. If you want to qualify tissue as a connective tissue, then it needs three components: some cells, fibers, and matrix. The matrix can be a solid, matrix can be semi-solid, matrix can be liquid. Okay. Now for loose or dense connective tissue, we will find semi-solid matrix, all the important cells and fibers. Okay. But for specialized connective tissue, maybe the matrix is present as hard matrix or liquid matrix, and some specialized kind of cells are present. Some specialized kind of cells are present into this particular tissue. That's why we have kept them into different group, that is specialized connective tissue, other than the cells which we have spoken about, right? Like you know that connective tissue has fibroblast cell, mast cell, plasma cells, lymphocyte cells, right? Adipocyte cells. But in bone, cartilage, lymph and blood, you will find some other types of tissue. Like in bone, you will find osteocyte. In cartilage, you will find chondrocyte. In blood, you will find RBC, WBC platelets. In lymph, you will find lymphocytes. So these are the different types of cell or specialized type of cell, which they have some specialized type of matrix. Even few specialized connective tissue, they don't have the fibers, right? So that's why they are very much special to us. Okay. Now let's talk about proper connective tissue or connective tissue proper or loose connective tissue. First, we'll talk about loose connective tissue. Now, why loose and dense? Now, the difference is the ratio or the proportion of the intercellular matrix and the fibers and cells. So, for loose connective tissue, there is more matrix and less fibers and cells. That's why the appearance is loose. Now, dense connective tissue, they have more fibers and cells, but less ground substance or less matrix. That's why the texture is dense, right? For specialized connective tissue, you will find special kind of cells present into them. Okay. Now, who will come under this umbrella of loose connective tissue? Now, if we talk about loose connective tissue, good example is areolar tissue and also adipose tissue. Areolar and adipose, they are examples of loose connective tissue, right? So areolar means, areolar came from the word area, and adipose, the word came from the word fat, right? Areolar tissue, the gaps, it's a gap filling kind of cementing tissue inside our body. Adipose tissue, the fat tissue. Okay. Now, if I'm talking about dense connective tissue, Again, dense connective tissue, they have certain types. Dense irregular connective tissue, dense regular connective tissue. Dense irregular connective tissue and dense regular connective tissue. Irregular connective tissue will find into the outer layering of different organs. Dense regular connective tissue, like their tendons, ligaments, will obviously we will learn in detail. Now, if I talk about specialized connective tissue, specialized connective tissue, if we talk about their types, there are two types. They are skeletal and they are fluid. Skeletal connective tissue and fluid connective tissue. Okay. Now, skeletal connective tissue, they will form the parts of skeleton or skeletal system, like their bones and cartilages. And for fluid connective tissue, blood and filtered blood or lymph, right? So, if you got my point, the difference between loose dense connective tissue and specialized connective tissue, we'll move forward, we'll learn in detail about the areolar tissue. Okay. So, when you talk about areolar tissue, as I said, we got the name from area. Means, if we talk about our hand, so we have bone inside that. Now, the bones will be covered by muscles. The muscles will be covered by skin. Right. But obviously, these are different types of tissue. Skin is epithelial tissue, muscle is muscular tissue, bone is a specialized connective tissue. What in between them? So, the tissue which will be filling the gap or cementing the empty space in between different tissue, and obviously it will make the framework of epithelial tissue. We will be calling the tissue areolar tissue. They're filling the empty areas in between different tissue, right? So that's why from the name area we got this term. So areolar tissue, they are also called as cementing tissue. They are forming framework of epithelial tissue. You know that epithelial tissue always rests upon connective tissue. So many epithelial tissue inside our body, they will be resting upon the areolar tissue. Okay. Now if I talk about example where you are gonna get this areolar tissue, they're present below the skin. Okay. So that was most of the things about areolar tissue. Basically, they're the gap filling tissue. Wherever you need to cementing, you need to fill up the gap with the help of biological cement, then our body will use areolar tissue to fill up the gaps. Like in, if we talk about plants, the same kind of like function is performed by parenchyma tissue, right? Now, what are the important cells of areolar tissue? Important cells of areolar tissue, we'll be finding fibroblast cells. Fibroblast cell, they are responsible for making the fibers. These white fibers are called collagens fiber. Collagens. Okay. These are the cells responsible for, these are the tissue where you are going to get macrophage. The phagocytic cells. This is the type of tissue where you are going to get neutrophils, the cells with multilobular nucleus. This is the tissue where you are going to get mast cells, cells which are responsible for secretion of chemicals like heparin, histamine, serotonin. This is the tissue you are gonna get cartwheel cells or plasma cells as well as few other lymphocytes. This is the tissue where you are gonna get adipocyte cells, fat cells, and this is the matrix. Inside the matrix also you will get these branched fibers of elastic fiber. You're gonna get the branched fiber of reticulin fiber, reticulin protein called reticular fiber. Right. So this diagram is also given in NCERT. So you're done with areolar tissue. Now we'll move forward towards another loose connective tissue. Do not forget that areolar tissue, that's a loose connective tissue. Next, we'll talk about adipose tissue. Okay, so when I'm talking about adipose tissue, that means fat forming and storing. So for fat formation and fat storage, you need particular cells. They are called adipocytes. This is the kind of areolar tissue. This is a kind of modified areolar tissue. Remember this. So areolar tissue is nothing but adipose tissue is nothing but modified areolar tissue. Right. They have many adipocytes cells, and these adipocyte cells, they're responsible for production of fat and storage of fat, right? Now, what is the function of adipose tissue? What is the function of fat, and where you're gonna get inside our body? Okay, again, you're gonna get below our skin, that is the subcutaneous fat. It is called hypodermis layer, right? Even the yellow bone marrow, that is also a kind of adipose tissue. Function is to provide insulation to our body. Insulation, fat is a bad or poor conductor of heat, that's why acting as an insulator. Also, it's acting as energy storage. 1 gram of fat, if you oxidize it, you are gonna get near about 9 kilocalorie of energy. So the adipose tissue that is responsible for energy as a tissue of energy storage, providing insulation to us, right? Okay, and also it protects many organs. Now, there are 2 types of adipocytes present into our body. Okay, one is called monolocular and another is called multilocular. There are 2 types of fat cells or adipocytes, right? Monolocular and multilocular. Now, to reveal their structure and function, we need to talk about them, right? Okay. So this is the nucleus. The nucleus generally present into the corner because the most of the space inside the cytoplasm that is occupied by the fat locule. There is a large globule of fat inside the cell. So if there is presence of only one fat locule or only one fat globule, that is called monolocular. And if there is presence of multiple fat particles, then that is called multilocular. Now let's talk about the distribution of mitochondria. Okay, monolocular fat has very less amount of mitochondria into them, but the multilocular fat cells, it has multiple number of mitochondria. It's rich in mitochondria, right? And this mitochondria inside, they will release a pigment called cytochrome, and this is also rich in iron. So that's why these fats, they will appear as brown color. So we also call this multilocular fat as brown fat. Brown fat. Okay. Now the fat cells or adipocytes with one fat locule, they have less mitochondria. So they appear white in color. So you also call them white fat. Okay. Now the fat present into adult body. So the fat of adult, they're kind of white fat. Now if I talk about brown fat, that is present in infants or toddlers. Okay, the fat present inside their body, there is a kind of brown fat. Okay, now where else we're gonna get this white fat? Okay, the blubber of penguins or the Arctic or Antarctic animals, like blubber of polar bear, seal, they're kind of also white fat. And the animals which will go for hibernation during the time of winter, that is called winter sleep. So they will accumulate brown fat inside their body and they will sleep throughout the winter months. So hibernating mammals or hibernating animals will accumulate brown fat into their body, but for human infants, they have brown fat. And the advantage of brown fat is that brown fat has more mitochondria, and as because there is more mitochondria, it will produce more molecules of ATP. So which fat will give more energy? Brown fat will give more energy, but the white fat or monolocular fat with monolocular adipocytes will give less energy comparatively to if you compare with brown fat, because they have less mitochondria, right? So that was all about adipocyte tissue. Okay. Now this was all about the loose connective tissue. Now let's learn about dense connective tissue. Okay. Now the dense connective tissue, there are of two types: dense regular and dense irregular. Now you all know that dense means there is more fibers and less matrix. That's why appearance is dense. Now what is regular and irregular? So it's all about distribution of those cells and fiber inside the matrix. If the cells or fibers are regularly distributed, parallelly distributed inside the matrix, we call that tissue dense regular connective tissue. If fibers and cells are not distributed in a proper manner, we call it dense irregular connective tissue. Let's find the difference. These are the cells and these are the fibers. See, they are properly distributed, they are properly arranged, nicely arranged into the matrix. This is the matrix. So into the matrix, cells and fibers, these are the fibers. Cells and fibers are nicely arranged into the matrix. So here, fibroblast cells and fibers are arranged properly into matrix. Okay. Now let's look into the dense irregular connective tissue. Okay. Now the cells of dense irregular connective tissue, they are not properly arranged. So as fibers So they are haphazardly arranged into the matrix. That's why we call them dense irregular, right? No proper arrangement, no proper arrangement of fibroblast cells and fibers. Okay, now let's talk about their examples. For dense regular, two important examples are tendons and ligaments, right? Okay, now tendons, they're responsible for joining muscle to bone. So tendons, see, so this dense connective tissue, they are also called as fibrous tissue, as because there is more number of fibers also present. These are also called as fibrous tissue as well, because more fibers are present here. So tendons, these are called white fibers. They connects bone to muscle. Ligaments, they're called yellow fibers as well. Just because there is more amount of elastic fiber present into ligaments, they join, so they connects bone to bone. Okay. These are muscles on bone. Now the attachment of bones to muscles will be there with the help of white fibrous tissue, that is tendons. Okay, as because the muscles have the elasticity, the tendons, they are inelastic in nature. They are inelastic in nature. Now the yellow fibers which are connecting bones to bones, we call them ligaments. That's because when you are moving the joints, you need to stretch the ligaments. So ligaments, the fibers of ligaments, they need elasticity, so they are elastic in nature. Sometimes you will hear in news that for this particular footballer or cricketer or sports person, the ligament is torn, so he'll be unable to play the match. So that's why the ligaments, they connects bone to bone. They connects bone to bone. That's why they're very crucial in terms of the movement of joint. But when you're talking about the connection of bone to muscle, you need to talk about the tendons, right? Okay. Now let's talk about some examples of dense irregular connective tissue. See, most of this kind of tissue, dense irregular connective tissue, along with epithelial tissue, they also will be forming the external or outer covering of many organs. Like for examples, capsule of kidney, example of dense irregular connective tissue, right? Okay. You know that epithelial tissue is responsible for formation of outer covering, but this tissue helps they will strengthen the outer covering along with the epithelial tissue, right? Gleason's capsule of liver. The outer covering of liver is called Gleason's capsule. Very important criteria of mammalian liver. Okay. Periosteum, the outer covering of bones. Perichondrium, outer covering of cartilage. All these are examples of dense irregular connective tissue. Okay. Now let's move forward to the next tissue, that is specialized connective tissue. First, we will talk about the skeletal tissue. Skeletal tissue means the two kind of skeletal components in our body, bone and cartilage. So let's talk about bone first. Okay. So when I am talking about bone, so this image you can see inside your imagination or inside your brain. Basically, this is a limb bone, right? Okay. So we have to explore the histological aspect of this particular bone. We have to learn the tissue present inside it, right? So when you talk about bone, there is obviously specialized connective tissue. Now, why is it specialized? Because it has specialized cells called osteocytes. The study of bone is called osteology. The study of bone is called osteology. Okay. So why it's specialized? Because of the special kind of matrix and cells. Okay. So when you talk about the cells present into bone, they are basically osteoblast cell. They are bone forming cells. They are responsible for formation of bone. Bone forming cells are called osteoblasts. And this osteoblast cell will get stabilized to form osteocytes. And another kind of cells are also present into bone. They are called osteoclast. They are bone eating cells. So you think that your bone is a static tissue, but actually it's not. Every time renovation is going inside your bones, osteoblast is forming the bones and osteoclast is demineralizing the bone. Every time recruitment of minerals will be there. Every time breakdown inside the tissue of bones will be there, right? But the stable cells you will be finding inside the bone, they are called osteocytes. So they are the main cells of bones. Remember there. Okay. Now this osteoblast will release some matrix and fibers as well. So if we talk about the fibers, we'll find collagen fiber into bones. If we talk about the matrix of bone, see, listen very carefully. The matrix is hard and non-pliable, means there is no bendability, stretchability inside the matrix. And due to the hard and non-pliability, the bone is rigid in nature. So bones are what? Bones are very rigid in nature, right? Okay. So due to the presence of this hard non-pliable matrix. Now we'll ask her, what is the forming tissue of this matrix? Forming components of this matrix. Okay. Matrix is made by inorganic salts like calcium salts and some organic components called oozine, right? So these are the important components of matrix of bone, calcium salts and oozine. Calcium salts means here specifically you will be getting calcium phosphate mainly, and also calcium carbonate, right? Okay, so that was all about bone. Now I'm coming to the tissue aspect of bone, histological aspect. Now how histologically bone is formed? The bone forming units, the structural units of bone, structural units of bones. Remember it, bones are of two types. Bones are of two types. The bones which is present outwards, that is having the rigidity, we call it compact bone. And the bone which is present inward, we call it spongy bone. So bone is of two types, compact bone and spongy bone. So if I am talking about compact bone, which is hard bone, the building unit of compact bone, we call it Haversian system or osteon, building units of compact bone. Now, if I am talking about the spongy bone, then the building unit will be trabeculae or honeycombs. They are honeycomb like structure, right? So these are the building units. So basically in NCERT the diagram is given, the diagrammatic representation, that is of osteon or Haversian system we need to study, right? So let's talk about this Haversian system or the building unit of compact bone. Okay, now what else you are gonna get inside the bone? The central part of bone, you will get the yellow bone marrow, written in shortcut YBM. That is basically yellow bone marrow. That is basically nothing but adipose tissue. In critical condition, this yellow bone marrow will take part, will get converted into red bone marrow and will help in formation of blood. But the red bone marrow that is present into the spongy bone region, RBM, that is responsible for blood formation or hemopoiesis. And in another chapter of locomotion and movement, we will study in detail different parts of bone. The apical part is called epiphysis, the neck part is called metaphysis, the middle part that is called diaphysis of bone. So there are different names for different parts of bone. And what is the zone where bones will exhibit the growth, that is called epiphyseal plate, that is present in between the epiphysis and metaphysis region, right? So everything we will study in detail in another chapter. The edge part we call it epiphysis, the body part is called diaphysis, the neck part is called metaphysis, right? Okay, so our topic is to learn about Haversian system of bone. So if you take a horizontal cut of bone and you observe under the microscope, you will observe the similar kind of structure. And this kind of cavities are present. This whole system is called one Haversian system, and in Haversian system multiple cavities are distributed and we call these cavities as lacunae. So what do we call these structures? Lacunae. Basically they are nothing but cavity like structure. Okay. What are these electron orbital like structure? We call these lamellae or Haversian lamellae. The central part is called Haversian canal. Okay. And we'll find special type of cells present inside the lacunae. Inside one lacunae, you will get one bone or matrix forming cell that is called one osteocytes. Inside one lacunae, you need to remember this is very important factor. The question can be asked, the very important thing, inside one lacunae of Haversian system, you will find one osteocyte. These osteocytes, basically they are modified osteoblasts responsible for formation of the matrix and fiber. This is one osteocyte inside one lacunae. This is the exclusive criteria of Haversian system present into the bone, right? So that was all about bone. Now we'll talk about cartilage. Okay, so the next specialized connective tissue of skeletal system, that is cartilage. Now we know that we have 206 bones present into our body. How many bones do we have? Adult human being, 206. But if you count the number of bones into infant one, then you will find that it's more than 300 bones. So along with our development with age, few bones will fuse together, they will merge together and they will form singular bones. Like example, our tailbone or coccyx. That is a fusion of 4 bones. The one another example is our sternum bone. That is fusion of 3 bones. Okay, so there are multiple bones you will be finding into our body. They are fusion of many bones. But if we talk about cartilage, they are physical entities, but there is no prominent number can be said regarding the cartilages. Yes. Okay. So some basic information about cartilages. They are also specialized connective tissue. Now regarding the cells of cartilage, we call them chondrocytes. They are modified chondroblast cell. The study of cartilage is called chondrology. Okay, the cells of cartilage, they are called chondrocyte cells. They are modified chondroblast cell. Okay, we know that epithelial tissue, they are example of avascular tissue. Yes, most of the cartilages, they are also avascular. Inside cartilage, there is no uh, entry or entry passage of blood or blood capillaries are there. Okay, so that's why we call that most of the cartilages are avascular. The blood capillaries will not enter inside the cartilage. It will remain like limited entry of blood capillaries is there with the outer covering of cartilage, that is called epicardium, right? Perichondrion. Outer covering of bone, periosteum. Outer covering of cartilage, perichondrion. Okay. Now fibers of cartilage, their collagen fiber. Matrix of cartilage. Okay, now cartilage matrix, due to the special type of matrix, cartilages are kept into the specialized connective tissue group, right? So when you talk about matrix, they have slightly pliable matrix which resist compression. So what do you mean by resistance against compression? Though cartilages are bendable, but you cannot depress them as you do with a sponge. They are not spongy in nature. So that's why they resist compression. Okay. Now what you will find inside the matrix of cartilage, that they will show this pliability. They don't have elastic fiber inside them. Okay, there are few cartilages where you will be finding elastic fiber, which we call yellow elastic cartilages, but there are many other types of cartilage like hyaline cartilage, right? You won't be getting much number of elastic or much amount of elastic fibers into them. So from where they are getting this pliability? Cartilage will get pliability from a special kind of salt called chondroitin salt. Like chondroitin sulfate, okay, and also one protein called chondrine. Now the diagram of cartilage as per the NCERT. Okay, what I was telling, the diagram of cartilage. So if this is a cartilage, there will be one outer covering. So that outer covering is also called as perichondrion. Right? Yes. Now there can be passage of blood capillaries up to perichondrion but not inside the cartilage, right? So that's why we say the most of the cartilages are avascular in nature. Now let's check inside. We'll find the matrix. And here also you will be finding some cavities. And you call these cavities as lacunae. But the difference is that unlike the lacunae of bone or Haversian system, there is no Haversian system. Remember this. There is no Haversian system present inside the cartilages, right? Now, inside the lacunae, you will find chondrocyte cells. Maybe one lacunae has one chondrocyte. You will find some lacunae with two chondrocytes. You will find some lacunae with four chondrocytes. You will find some lacunae with three chondrocytes. So, the number of chondrocyte inside lacunae is one or more than one. 1, 2, 3 or 4. Inside one lacunae, how many chondrocyte? 1, 2, 4 chondrocyte in one lacunae. So that is the difference between the histological aspect of tissue of bone and cartilage. So in NCERT, if you follow the diagrams of cartilage and bone, the histological diagram of bones and cartilage, you will find they are quite confusing. But what you have to calculate? You have to calculate the number of osteocyte in one lacunae and the numbers of chondrocytes inside one lacunae, right? And in cartilage, the structure of cartilage, there is no presence of Haversian system like which we found inside the bones. So basically, this is the structure of one Haversian unit or one osteon, one Haversian system. There is thousands of Haversian systems are present inside the compact bone, right? Okay. Now, the function of bone is also asked. The function is very important. You need to remember all the functions of bone. First, form the framework of body. Forms the framework of body. Based upon the framework, that is the skeletal framework, the body design can be imagined, right? So the skeleton or the framework is very much important. Okay. Then the next function also carry the weight of our body, specifically our limb bones. Carry the weight of our body. Bones are also responsible for hemopoiesis, right? Under the influence of erythropoietin inside the red bone marrow, blood formation will take place. There is the process of blood formation is called hemopoiesis or hematopoiesis. Okay. Bones also helps us to maintain the balance of mineral into our body, maintain mineral balance, right? So if you demineralize bone, what you will get? You will get calcium ions, you will get phosphate ions, right? So maintaining the osmotic balance is also important for us, and for that we need to maintain the mineralic balance. So according to our need of our body, the demineralization and mineralization of bone will take place, and obviously few hormones will be influencing those things, right? There the hormones of called uh, thyrocalcitonin, the hormones called parathyroid hormones, the hormones called calcitriol or vitamin D. So these are different hormones will take place in to this mineralization and demineralization of bones to maintain the osmotic balance of our body. Okay, and also another function of bone, bone protects many vital organs like our skull is protecting our brain, few bones protecting our kidneys. Okay, so bones also protect our heart, the rib bones protect our heart and lungs. So vital organs are also protected by bones. Also, bone forms joints and along with the help of muscular tissue help in locomotion and movement. Okay, so we are done with skeletal connective tissue. Now we'll talk about fluid connective tissue. See, fluid connective tissue, they're also called as false connective tissue, they're also called as afibrous connective tissue, a fibrous connective tissue. Now why false and why a fibrous connective tissue? Bone and blood and lymph, fluid connective tissue, we have to talk about blood, we have to talk about lymph, right? So why blood and lymph are false connective tissue? Is because the matrix of blood and the lymph, which is called plasma, the matrix, the liquid matrix of blood and lymph, that is called plasma, that is not made by the cells of blood or lymph, that is produced by other organs like liver or spleen. So as because for this specialized connective tissue, the matrix or the ground substance, which is liquid or fluid in nature, is not produced by its own cell, is produced by some other gland or some other organs like liver or spleen. That's why we call this connective tissue as false connective tissue. And that's because there is no trace of fibers, right? There is no collagen fiber, elastic fiber or reticular fiber present inside the blood and lymph. They're called afibrous or afibrous connective tissue. Got it? Okay. Apart from this plasma, blood also has the formed elements. The cells of blood which are formed inside the bone marrow and then released into the stream, bloodstream. That's why we call these cells as formed elements. Okay. They are red blood corpuscles, white blood corpuscles and platelets. Okay. Now remember that platelets are not true cells. Platelets are not true cells. Platelets are broken pieces of another cell called megakaryocyte cells. So platelets, basically the meaning of platelet is broken pieces. So if someone asks you which components of blood is or are not true cells, they don't have nucleus or they don't have any other cell organelles, they are platelets. Okay. For mammalian RBC, when they are matured, they will lose their nucleus, mitochondria and Golgi complex. So that's why we call them enucleated RBC. But non-mammalian RBC, they are nucleated in matured form. If someone asks you what about immature RBC or erythroblast, they have nucleus. But mature mammalian RBC, they don't have nucleus, right? Okay. So in detail, we'll learn already we have learned in the chapter of body fluids and circulation. Okay. Now what about lymph? Lymph is called as filtered blood. Lymph is also called as middleman of the body because lymph connects blood to tissue. Blood circulates inside a closed circulatory system which is connecting the tissue with the help of lymph. That's why you are calling it middleman. And inside our tissue, through the capillaries, few components of blood will get filtered out and will form colorless fluid called lymph. So basically lymph has plasma and WBC. Lymph is plasma plus WBC, and specifically the lymphocytes of WBC. Lymph is rich in lymphocyte WBC. That's why this colorless fluid, which is filtered blood, also called as tissue fluid. You will find this into the interstitial space, right, into the tissue. That's why we call it tissue fluid. So difference between blood and lymph: blood has components like blood and formed elements, plasma and formed elements. Plasma is the liquid matrix. The component of plasma of blood and lymph is more or less same, but few proteins will be finding extra into the plasma. Few proteins, basically the few clotting factors, okay, they are present in very less amount or in very marginal amount into the plasma of lymph. Otherwise the osmotic balance, right, the tonicity of plasma of blood and plasma of lymph is similar. Okay. Now blood has the formed elements like RBC, WBC and platelets. Lymph, RBC is absent. Platelets also very marginal amount you may find inside the lymph, but you will be finding good numbers of lymphocytic WBC inside this colorless fluid. Blood is red in color inside our body, lymph is colorless. So with this we can conclude the connective tissue. Now we will move forward to the muscular tissue. So let's solve few questions and we will move forward to the next topic, muscular tissue. Okay, so the question is given here: choose the correctly matched pair. So tendon, they are specialized connective tissue. Are they? They are not specialized connective tissue. They are dense connective tissue. Adipose tissue, are they dense connective tissue? No, they are loose connective tissue. Areolar tissue, yes, areolar tissue. Are they loose connective tissue? Exactly. So this is our answer. Areolar tissue is perfect match for loose connective tissue. Cartilage, they are also loose, not loose connective tissue. Cartilages are specialized connective tissue. Okay. Now the next question: the kind of tissue that forms the supportive structure in our pinna, that is external ears, is also found in tip of nose. Exactly, not into the vertebrae, not into the nails, not into the ear ossicles. These all structures, vertebrae and ear ossicles, they are bone, and nails, basically they are keratinized compound, keratinized epithelial tissue. Okay, so tip of nose also has the cartilage as well as our ear pinna. Okay, Which of the following is the most abundant protein in animal? Okay. Remember, we were talking about collagen fiber, which was secreted by fibroblast cell. So collagen fiber is made up of collagen protein, and basically you can correlate this topic from the chapter biomolecules. In that chapter you will get the answer. Collagen is most abundant protein in animals, not most abundant protein in biosphere. That is a different thing. Most abundant protein in biosphere, that is one of the enzyme, that is rubisco enzyme, right? Rubisco is most abundant protein, but most abundant protein in animal kingdom, that is collagen. Next question, which of the following is present between the adjacent bones of the vertebral column? Okay, so if we talk about the adjacent bones of vertebral column, cartilaginous tissue will get, that is intervertebral disc, that is called intervertebral disc. So basically that's a cartilage. So this is our answer. This is our answer. Intercalated disc. This particular structure will find inside the into the cardiac muscle. So do not confuse between intervertebral disc, which is cartilaginous tissue, intercalated disc. Intercalated disc, that is a different set of tissue that is present in cardiac muscle, which is basically gap junction. Okay. Now we'll talk about muscular tissue. Muscles, they are tissue which are responsible for movement and locomotion. Tissue responsible for movement and locomotion. Yes, muscles are mesodermal in origin, except the muscle, few muscles present into eye, not all eye muscle, that is the iridial muscles present in iris, and the ciliary body muscles present inside our eyes. Okay, the muscles which are responsible for eyeball movement, they are mesodermal in origin, but I said the iridial muscle of iris, and ciliary body muscles, again as associated muscles with our lens of our inside our eye, their origin is ectodermal in origin. Remember this, they're exceptional. Okay, mesodermal except iris muscle and ciliary body muscles. Okay, muscles are also responsible for heat production and force generation. Muscles are also responsible for, muscular tissue is responsible for heat production. So that's why when our body is feeling cold, body temperature is going below 37 degrees Celsius, our body starts shivering. Rapid contraction and relaxation of muscles, and when there will be rapid contraction of muscle, it will produce heat. Okay, so heat production is also one of the important function of muscular tissue. That's why when you are doing a heavy physical work, or when you are doing gym, or when you are running, you will start sweating after some time, right? Okay, so we have to talk about the different types of muscle. We need to classify different types of muscle. So when you talk about types, muscle are of three types: skeletal muscle, visceral muscle, and cardiac muscle, based upon their location. Muscles are of three types based upon their location: skeletal, visceral, and cardiac muscle. Okay. Now, based upon their appearance, let's check their diagrams. They are slender, cylindrical shaped muscles with multiple nucleus, multinucleated cells. Basically, the skeletal muscle cells are multinucleated. You will find dark and light banding, that is also called striation, into this particular muscle. That's why skeletal muscles are also called as striated muscle. So this dark light banding, that is called striation. Dark band, light band. Sorry, dark band, light band. 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 That is called striation. So that's why the skeletal muscle cell tissue is called also striated muscle, multinucleated muscle, and cylindrical cells. Shape is like cylinder, that's why we are calling it cylindrical muscles. These are multiple nucleus, many nuclei. Plural form of nucleus will be nuclei, right? And they are under our voluntary control, direct controlled by our central nervous system or central nervous system, direct control. That's why we call it voluntary muscle, under voluntary control, under voluntary control, right, of somatic branch or somatic nerves. Means this muscle will listen to you. This muscle will listen to you. You can contract and relax them upon your will and wish. That's why we are calling them voluntary muscles. Okay. Now, there will be very fast or very quick contraction, powerful contraction will be there. That's why easily they will get fatigue. They will get exhaust. So they're also called as fatigue muscle. They will get exhausted very soon. Fatigue is observed into these particular muscles, right? Now, if we talk about visceral muscles, their cells, they will look different. The spindle shaped cells with single nucleus. There is no banding pattern is observed. That's why this is also called as non-striated, or you can call it smooth muscles. Now the skeletal muscles, they are present on skeleton. They are present on bones associated with skeleton. Right. But visceral muscle, they are not associated with skeleton, but they are present into the inner lining of, the middle lining of visceral organs. They are present into the middle lining of visceral organs. This red part is basically muscular part. So that's why internal organs are called viscera. There you will find this visceral muscle into the wall of hollow organs, right? Like inside your blood vessels, inside your stomach, inside your intestine, the muscular layer that basically made by visceral muscle. Okay, there is uninucleated. See, the cells are, the shape of the cell is not like cylinder. The shape is called fusiform cell due to the presence of this tapered end. Fusiform or spindle shaped cells are present. No striation is found. No striation is there. And obviously they are not under your voluntary control. That's why they are called as involuntary muscle. They are not under your voluntary control, right? They are called involuntary muscles. Now cardiac muscles, they're present into heart, into myocardium of heart. You'll find cardiac muscle into myocardium layer of heart. How they will look like? They are branched cells with single nucleus but with striation. So they are branch striated muscles. This is one cardiac muscle cell. Now I am drawing another cardiac muscle cell. This is the nucleus. And this is the striation. Okay. So these cells are branched cells. This exclusive feature of cardiac muscle cells. None of the other muscular cells, either you talk about skeletal muscle cell or visceral muscle cells, they don't have branches. They are uninucleated. They have, one cell has only one nucleus, but striation is present. You can find the striation. Striation is seen. So branch striated means cardiac muscle cells. Unbranched striated means skeletal muscle cells. Multinucleated striated means skeletal muscle cell. Uninucleated striated means cardiac muscle cells. So the concept, you need to learn the concept in a very well manner, right? So whenever they are asking for assertion and reason or statement based question, the sentences can be modified, right? So what I said, so both are striated, but what are the basic differences in between them? Okay, now if we talk about the control, skeletal muscle cells are under voluntary control, but cardiac muscle cells are involuntary in nature. So there also can be called as involuntary striated cells. They're involuntary in nature, right? Okay. Now a special structure you are gonna get, at the junction of two cardiac muscle cell to pass the message from one cell to another cell. Basically, a disc kind of structure is formed at the junction of two cardiac muscle cell, and the disc is called as intercalated disc. Intercalated disc, in some previous question we were talking about this intercalated disc that is present into cardiac muscle cell at the junction of two branched cell or two cardiac muscle cell. They will get jointed or conjoined with the help of a disc like structure. Basically this disc is helping the communication between two cells with the help of gap junctions. So intercalated discs, they are nothing but a kind of cellular junction, and basically gap junction is present here. They help in cellular communication. Remember this, gap junctions, they're responsible for intercellular communication. So this intercalated disc, they are nothing but gap junctions. So I think the differences are clear now in between the skeletal muscle cell, visceral muscle cells, and cardiac muscle cells. Okay. Now few more aspect of muscular tissue. Again, there will be in detail elaborated discussion on muscle in the chapter locomotion and movement. In that chapter, there will be elaboration of bones, cartilage, and muscular tissue. So this is a brief flash of muscular tissue. So when you talk about muscles, the important properties of muscles, you're gonna get excitability, contractility, extensibility, elasticity, and conductivity. Now basically, whenever there is movement in muscular tissue, there will be change in the size of muscle. Like if you are moving your biceps, whenever you are showcasing it, the size of bicep muscle, the muscle is bicep muscle, that will decrease. There is a kind of voluntary muscle, right? Whenever you are stretching your hand like this, there will be elongation, extension of your bicep muscle will be there, and after every contraction or extension, the muscle will get back to the original position, that is called relaxation, right? So excitability, in order to give the signal, yes, muscle, you please start contraction and relaxation, you need to give some voluntary signal or involuntary signal, right? So that signal can be sensed by a particular cell, and the capability of sensing the neural signal, that is called excitability. Shortening of muscle is called contractility. Lengthening of muscle is called extensibility. The capability of shortening and lengthening and again coming back to the original position, that is called elasticity of muscle. And conductivity, the muscle cells have the capability to conduct that nerve signal within the like sarcolemma or the cell membrane of muscle cells, right? So the study of muscle, we call it myology or sarcology. Myology or sarcology means study of flesh. So flesh in our body is mainly made by muscles, right? Okay. And the muscle forming cells are called myoblast. Muscle forming cells are called myoblast cell. No, I was talking about skeletal muscle cell. So where you are gonna get skeletal muscle cell, or some examples of skeletal muscle cell, like bicep, triceps, right? The limb, the muscles which are associated with our limbs, all of them are voluntary muscles or skeletal muscle, right? So these skeletal muscle cells are multinucleated. Now, how this multinucleation is happening into them? So suppose this is a myoblast cell, Now primarily they are uninucleated. There is another myoblast cell. There is another myoblast cell. So what will happen when the muscular development is happening inside our body, or embryonic development or morphogenesis is occurring inside? So the cell will get a urge, a need to share their resources. That's why they will get fused with each other. So there will be a fusion of many or multiple myoblast cells, and they will fuse together to form a muscle cell or muscle fiber which has multiple nucleus. That's why there is the origin of multinucleated muscle cell or muscle fibers. Muscle fibers are also called as muscle cells. Muscle cell equals to muscle fiber. One muscle cell equal to one muscle fiber. Okay. Now let's elaborate this contractility of muscle. So what you can observe here, this is contraction of muscle. Contraction means shortening. Shortening of muscle is called contractility. Lengthening of muscle, elongation, beyond the normal range of muscle, or the lengthening of muscle is called extensibility. And either they are going for contraction, they are going to get back to the original structure or original size of muscle, that is called relaxation. And the property is called elasticity. Right? You need to remember this thing. These are very much conceptual thing to understand. Right? Relaxed stage means the normal size of muscle. Shortening of muscle, contraction. Extending or lengthening beyond the range, that is called extension. Okay. Now let's move to the next type of tissue. There is neural tissue, and after neural tissue we'll talk about different type of cell junction, and we'll practice some important question, and then we'll end this particular chapter and our one shot will be completed then, right? Okay. Now before learning the neural tissue, few more important thing we need to talk about muscles or muscular tissue. So basically what happens, we need to talk about one single muscle that is made up of numerous muscle fibers or muscle cells. One muscle has many muscle fiber or muscle cells, right? Now, if you get inside the muscle fibers, you will find that tiny, like inside one muscle cell, some unit of contractions are present on a tiny fibril kind of structure that is called myofibrils. So each muscle fiber has multiple myofibrils, and each myofibrils have multiple myofilaments. One muscle fiber has many myofibrils, and one myofibril has myofilaments. These are basically kind of protein which are responsible for contraction and relaxation of muscle, and these myofilaments are called as actin and myosin. These are called myofilaments or contractile proteins, right? So in detail you are gonna learn into another chapter where you will learn very interesting topics of muscle contraction, the sliding filament theory and all. Right. So if this is a bone, with the bone muscle is attached. If you are eating chicken leg piece, so this is whole muscle, and that also has a covering, right? And the connecting tissue between muscles and bones, they are what? Please comment in the box. The connective fibrous tissue which connects bone to muscles, they are called tendons, and inside one single muscle you will find multiple muscle bundle or fascicles. So multiple fascicles are present. What are these fascicles? Fascicles are nothing but bundle of muscle fibers. So these are fascicles. Now inside fascicles you will get muscle fiber. Now if you check one muscle fiber, inside that muscle fiber you will get myofilaments which has striped pattern. Now if you check one myofilaments, you will get this myofibrils. Inside this myofibrils you will get actin and myosin, right? Now sometimes the covering is also asked. The immediate covering of one muscle cell or one muscle fiber is called endomysium. Covering of each muscle bundle or fascicle is called perimysium, and the external covering is called epimysium. So this is all about muscle. This is a single muscle. This is muscle bundle of fascicle, and fascicle is made up of muscle cell. So you can note it down in a proper manner. Right? Okay. Now if you are done with it, one more question can be asked. Now this muscle fascicles, These are connected by connective tissue, and we are calling this as fascia, right? So if this is one muscle fiber or muscle cell, and if you take many muscle fibers together, we will call it muscle bundle or fascicle. Now, if you take such muscle bundles, many of such muscle bundles to make a single muscle, you need to connect them, you need to join them with the help of connective tissue, and that connective tissue is called fascia. So, remember the difference between fascicle and fascia. Fascicle is the bundle of muscle fibers or muscle cell, and the tissue which helps them to combine together to form a muscle, that connective tissue is called fascia. Now let's talk about neural tissue. Okay, so neural tissue basically is made of by two kind of cells, neurons and neuroglia, right? So neural tissue is responsible for sensitivity, responsible for control and coordination. Controls all the activities of our body, either its movement, either the voluntary activities or the peristalsis of your food pipe, peristalsis of your gastrointestinal tract, or the involuntary movements happening inside our body. Everything will be controlled by our neural system or nervous system, and the tissue is called neural tissue, right? Okay. Now, histological, we have to only discuss the histological aspect of neural tissue in detail. We are going to learn in another chapter, neural control and coordination, right? Okay, so neural tissue, that is mainly made of by the structure and functional units called neurons, and also another type of cells or helper cells, that is neuroglia. Neuron and neuroglial cells, the structural units of nervous system, neurons and neuroglia, and structural as well as functional unit of nervous system, they are neurons, right? So basically, what is the function of neurons and neuroglia? In your home, if you observe that there are conducting wires through which electricity will pass, right? Primarily they are made up of copper or mixture of copper or silver, right? Because they have good conductivity. Now, if you directly touch that copper wire, then there will be damage into your skin or into that copper wire. So, in order to protect the copper wire and to protect you against the damages or against getting shocks, electric shocks, these are covered by Teflon or insulator kind of polyethylene, right? So this insulating covering and the copper are forming the electric wirings of our body. Similarly, of a home. Similarly, the electric wire, they are acting like neurons, and their plastic coverings, they are acting like neuroglia. Clear? Is it clear? So neurons, they are responsible for sensing the signal. Every time our body is getting signals, we call them stimuli. The stimuli or the stimulus can be external or internal, and the function of the neuron is to sense them, to classify them, to conduct them, right? The function of neurons sense stimuli, conduct the signal, right? Sensing and conducting. So basically what they're sensing, neurons are sensing stimulus in singular, stimuli in plural, and what they're conducting, they're conducting nerve impulse. As soon as they will get that stimulus, suddenly, instantly, they will convert that stimulus into electrochemical signal that we call nerve impulse. Electrochemical signal. In detail, how to conduct this nerve impulse, how nerve impulse is conducted through a single neuron, how the nerve impulse is transmitted from one neuron to another neuron. In detail, you are going to learn in the chapter control and coordination, neural control and coordination. Here we will be only talking about different types of neurons and the structure of neurons, different types of neuroglia and their structures and function. Okay. When I'm talking about neuroglia, basically they are helper cells. They're providing nourishment to whom? Providing nourishment to neurons. They also forming the blood-brain barrier. These neuroglial cells are responsible for filtering blood and providing only selective nutrients to the tissue of our brain. It is called blood-brain barrier. This neuroglia also responsible for phagocytosis, special kind of neuroglia are present into our nervous tissue. This nervous tissue, you know, they are ectodermal in origin, but special type of phagocytic cells are present into these neuroglial cells as a part of the neuroglia in our nervous system. They are mesodermal in origin, exception of neural tissue. Most of the neural cells are ectodermal in origin except this phagocytic neuroglias. We call them microglia. They act as phagocytic WBC, they act as macrophages. Basically, they are modified macrophages. So basically, what is their function? Their function is to clear the dart particles or debris from our central nervous system. Okay, and also sometimes the neuroglia, they help the neurons for damage repair. So repairing neurons, helping neurons, insulating neurons. And providing nourishment to the neurons. Multiple functions are taken care of by these neuroglial cells. Okay. Now, when you talk about neuroglia, in our central nervous system, you will find few types of neuroglia, that is CNS. In our peripheral nervous system, you will find few types of neuroglia. Remember, these are not CNS and PNS, these are not types of neuroglia. We are going to learn now that in central nervous system, what kind of neuroglia you are going to get. Yes, in central nervous system, you are going to get oligodendrocytes, you are going to get astrocytes, you are going to get microglia, you are going to get ependymal cells. So these are different types of neuroglial cells you will be finding into the central nervous system. In peripheral nervous system, the different types of neuroglial cells you are going to get Schwann cell, satellite cells. Okay. Now, what about their functions? Yes. Oligodendrocytes and Schwann cells. Oligodendrocytes in central nervous system, Schwann cells in peripheral nervous system. They are responsible for formation of myelin covering that we call myelin sheath, outside covering of the axon of a neuron, right? So that is called myelination. Formation of myelin sheath, right? Astrocyte, they're responsible for forming blood-brain barrier, BBB, blood-brain barrier. This is a function of astrocyte. Also, they help in damage repairing purpose. Microglia, they are the phagocytic cells. Microglia, I am saying that these are the imposter cells, right? These are mesodermal in origin. Most all other cells are, including neurons, astrocyte, oligodendrocyte, swan cells, satellite cell, ependymal cells, all are ectodermal in origin. Why it's imposter? Because it is mesodermal in origin, acting like cell of connective tissue, acting like macrophage. Okay. Now we'll talk about neurons. Now, what are the different types of neurons we are gonna get inside our nervous system? Okay. If we talk about the neuron types, they are afferent or sensory neurons, they are efferent or motor neurons, they are intermediate or relay neurons or interneurons. So these are three types of neurons based upon their function: afferent, efferent or relay. Or afferent neurons are called sensory neurons, efferent neurons are called motor neurons, relay neurons are called interneurons. So basically what happens, our body has sense organs, they are acting like a receptor of stimuli. So suppose inside your eyes you have retina, right? So the cells of retina, they will act as the receptor of light. So the light ray will be sensed by the receptor cells. So what we call? We call these receptors. Now this light signal will get converted into electrochemical signal, and we have some special optic neurons which are part of optic nerve. They will conduct this impulse and it will take to our processing area of our brain. That is the headquarter of our central nervous system. The processing unit of our body is present into our brain. So the neurons which are responsible for conducting the nerve impulse from receptor to CNS, they are called sensory neurons. Okay. Now your brain will process the data. Now your brain will give some output. Now the neurons which are responsible for carrying the output from brain towards the effector. These neurons are called motor neuron. Now who are acting as effector? There are glands and muscles, right? Like for example, if there is a source of bright light, I'm putting towards my I'm putting towards my eye. So what will happen? Suddenly I will close my eye. Why so? Because the brain has processed the data. The intensity of the light rays are quite higher, so you need to contract your muscles of eyelid. So that's why you will blink suddenly. So eyelid muscles are acting as an effector, and the signal is sent to your eyelid muscles from your brain with the help of motor neurons, and the light ray, they acted as the stimulus here, right? Okay, so that's how different neurons will work accordingly. So now uh, we'll learn the structure of a neuron. Now based upon the structures also, neuron can be of different types. Few neurons, they have only one cell body. We call this kind of neurons as apolar neuron. Neuron, they're responsible for providing sense. They will sense a signal and they will transmit the signal. For sensing and transmitting, they have different branches associated with the cell of neuron. We call those branches as dendrites and axon. So if you get a neuron which has one dendrite or one dendron and one axon, we call it bipolar neuron, right? You will be getting this kind of neurons into your eyes, into your ears, into your nose. The neuron which has only one arm, only axon, we call it unipolar neuron. Unipolar neuron are found into our embryonic developmental period, and gradually they will be developed either into bipolar neuron or into multipolar neuron. Neuron with multiple dendron and one axon, that is called multipolar neuron. Neurons of our cerebral cortex, they are multipolar neurons. And a special type of neuron with one cell body, one dendron and one axon. It has one dendron, the short process or the short arm, the long arm called axon. So technically it has two arms, two poles, one dendron and one axon. But as because these poles are present into a single axis, in under microscope, when you are observing, you will think that this particular neuron has only one pole or one arm. So that's why we are calling it pseudo unipolar neuron. And this pseudo unipolar neuron you will be finding into the sensory arms of our spinal nerves. Okay, so in detail, obviously everything you are gonna learn into the chapter of neural control and coordination. Okay, now the structure, if I talk about the structure of neuron, So this is the cell body of a neuron. Cell body. What's happening? Or soma or cyton or neurocyton. Okay. This is the nucleus of the neuron. This is the cytoplasm of neuron. This is the cell membrane of neuron. And neurons, they're rich in mitochondria. It's because nerve impulse propagation needs a lot of energy, right? We'll be finding a lot of mitochondria and also bunch of ribosomes which are grayish in color. We call it Nissls granule. Or Nissls bodies. Basically, these are nothing but ribosome help in protein synthesis. Okay. Now these short processes or these short arms are called dendrites. This long arm is called axon. The point where axon has started, that is called trigger zone or axon hillock. The cytoplasm inside the axon is called exoplasm. The cell membrane of axon is called exolemma. And the end part of the axon, that is called axon terminal. The button-like structures, basically, it has some vesicles of neurotransmitter. We call this structure as presynaptic knob. These presynaptic knobs, they are basically a reservoir of neurotransmitter in the form of inside the vesicles present inside them, right? Okay. Now this is a neuron, and this is also called as non-myelinated neuron. The neuron, the axon part also has some cellular support called Schwann cell or oligodendrocytic cells. So the cells surrounding the axon, we call them Schwann cell. If this is a neuron of peripheral nervous system, if this is a neuron of central nervous system, instead of Schwann cell, you will be getting what? You will be getting oligodendrocyte. Now, how these cells are covering the neurons? So this is my headphone. Suppose this is a Schwann cell. Yes, the Schwann cells are elongated cell. So like this, they will wrap the length of axon, right? So either the neuron is myelinated or non-myelinated. The axon is wrapped by Schwann cells in peripheral nervous system and oligodendrocyte cells into central nervous system. Okay. Now the cell membrane of these Schwann cells are called neurolemma. Do not confuse between axolemma and neurolemma. right? And sometime what happens, sometime these Schwann cells or oligodendrocyte cells, they will secrete white colored sphingomyelin, special kind of lipid to wrap certain portion of axon or axolemma. This white colored sphingomyelin, we call it myelin sheath. So myelin sheath, this myelin basically made up of what? Made up of a special kind of lipid, we call it sphingolipid or sphingomyelin. Okay. So based upon the presence of myelin sheath, neurons again can be of two type: neuron with myelin sheath, neuron without myelin sheath. Neuron with myelin sheath are called myelinated neuron. Neuron without myelin sheath are called non-myelinated neuron. Non-myelinated neuron. And the zone where in myelinated neuron, if I am talking about myelinated neuron, the zone where myelin sheath is not present, that is called node of Ranvier. So node of Ranvier, how you can define it? The non-myelinated part on a myelinated neuron. Non-myelinated part on a myelinated neuron. What is the difference between this myelinated neuron and non-myelinated neuron? Remember, if I talk about the speed of nerve impulse propagation, the dendrites act as the afferent arm of neuron. They receive the nerve signal. Dendrites act as the receiver part of nerve signal. Okay. Axon act as the conducting part of this nerve impulse, and presynaptic nerve, they act as the delivering part of this nerve impulse to the next neuron. So if someone asks you which is the afferent arm of neuron, afferent means receiving. Which is the efferent arm? Efferent means donating arm. So neuron is receiving nerve signal with dendrites. Neuron is donating nerve signal or conducting nerve signal with the help of axon, right? Can you remember it? And the cell body acting as a relay part, is connecting the dendron and axon, right? And at the place of axon hillock, the nerve impulse formation is triggered. That's why this is also called as triggered zone. Okay. Now, when the nerve impulse is propagating with the axolemma, the membrane of axon, if it's myelinated, the nerve impulse will propagate as a jumping manner. That is called nerve impulse saltation. Okay. If the axon has no myelin sheath, in normal continuous way, the nerve impulse will propagate. So if you are walking and beside you someone is jumping, so who will move faster? Obviously, the person who is jumping. So in myelinated neuron, due to the jumping pattern or the saltation pattern of nerve impulse, the myelinated neurons propagate or conduct the nerve impulse in a very fast manner, as well as but in case non-myelinated neuron, comparatively the propagation speed is slower. Okay, now we'll talk about different type of cell junctions. So this is gonna be the last topic of the chapter, structural organization in animals, and the topic is animal tissue. The last topic to discuss, cell junctions. Yes, because cell junctions, when you talk about this particular structure, they are intercellular junctions. They are intercellular structures, means present between two cells. Why they are present? They help the cell to get properly attached. So they will form a firm attachment between the cells. So they are responsible for providing the mechanical strength. They are responsible for providing mechanical strength between the attachment of two cells, because if you want to form a successful, good, a strong tissue, okay, you need to like stop entry of few unwanted things, right? For that, proper attachment between the cells are needed. Also, they help the cells to communicate. So mechanical strength and providing communication, providing mechanical strength and providing communication between cells, that is the function of cell junctions. Yes. Now very quickly we will be finishing the topic. In NCERT, cell junctions are of, according to NCERT, three types: tight junctions, tight junction, adhering junctions, and gap junction. Tight junction, gap junction, adhering junction. Okay. Tight junction and adhering junction, these are the junctions responsible for providing mechanical strength. Gap junction is basically communicating junction. Okay. Now, in one sentence, if I want to explain their function, for tight junction, that prevent leakage of substance into a tissue. If I want to talk about adhering junction, the function of the adhering junction, they're the strongest junction in between two cells. Okay, though in detail, if you want to learn in detail, there is another type of junction you will be finding that is called desmosomes, right? So basically in NCERT you can consider these desmosomes and adhering junctions, they are kind of similar, So adhering junctions, basically they are called cementing junctions, or their function is welding, formation of a weld, a strong attachment of two cells. Okay. Gap junctions, communication. Gap junctions help in communication. Basically they are intercytoplasmic bridge between two cells, transfer of ions, signals, small or large molecules will take place with the help of a bridge. I'm saying the cytoplasmic bridge between two cells, and this bridge are called connection, not connection. These bridges are called connection, because basically multiple units of connection will form a gap junction, right? Okay. So this kind of question can be asked based upon their function, right? So tight junction, if this is a cell, suppose this is one cell and this is another cell. This is the nucleus of them, and like sewing, you know, when you are stitching the cloths, you have to sew it. So if you are stitching the cell membrane of two cells like sewing, and that junction you need to be placed on the apical part, the apex region of two cells. Towards the apex, you will find this kind of junction, and that is basically tight junction. That will stop leakage of substance. Okay. Now, at the middle part of two cells, the kind of welding junctions with the help of special proteins will be made, that is called adhering junctions. You don't need to learn about the specific name of the proteins. In higher classes, you can learn in detail, right? Okay. So the cementing or welding junction like this, two cells will be attached together with the help of adhering junctions. This is adhering junctions. And if there is tunnels or connection through which a cell can pass the cytoplasmic components from one cell to another cell, so that can be called as gap junction. A very famous examples of gap junctions, they are intercalated disc. Intercalated disc of your cardiac muscle, they're a very good example of gap junctions. Even the electrical synapse, in detail, when you are going to line the neuron to neuron junction, that we call synapse, we have two types, chemical synapse and electrical synapse. Chemical synapse, in between two neurons, a junction is there where there will be a gap or cleft, that is called chemical synapse. When there is very minimal gap or there is no gap between two neurons, that is called electrical synapse. So when there is electrical synapse, the nerve impulse will be propagated from one neuron to another neuron with the help of gap junctions. Right? So again I am repeating these three kind of cell junctions. Remember all these kind of junctions where you are gonna get? You are gonna get inside the epithelial tissue. Muscular tissue, you will get gap junctions into them. Okay. Tight junctions, basically this is the junction responsible for blocking the leakage, right? For example, if you observe the inner wall of your stomach, if you observe the inner wall of stomach, special cell or the epithelial cells of stomach, they will release HCl into the lumen of stomach. Now there is a chance that the HCl can damage the tissue. So for that, the first precaution is the inner lining of stomach is protected with the help of a layer of mucus. Further, in between the cells, you will be finding tight junction that is stopping the entry of HCl in case of gastric ulcer and those diseases. So the tissue or the inner lining of stomachs cannot be fully disturbed or fully damaged with the help of with HCl. So it's due to the mucus layer and tight junction present into the inner lining of stomach, the cells of stomach, the HCl cannot affect them, right? So that is the importance of tight junction, and you can imagine how it's happening and how it's helping the cells to stop the leakage of HCl inside the tissue. When you're talking about attaching the two cells with a firm connection, that is called adhering junction, acting like welding. And when you are talking about cytoplasmic connection or communication between two cells, obviously we need to talk about gap junctions. Okay, now with some questions, let's try our understanding. Okay, so identify the types of cell junctions that help to stop the leakage, okay, of the substances across the tissue and facilitation of communication with neighboring cells. So we have to find two junctions, and our answer is type of cell junction help to stop the leakage, that is tight junction, and communication means gap junction. So our answer will be option 2, tight junction and gap junction respectively. Okay. Next question, mast cell secrete. Mast cell, three component is secreted: heparin, histamine and serotonin. Hippurine, no. Myoglobin, no. Histamine, mast cell secrete histamine. Hemoglobin is not secreted by mast cell. So we got our answer. Now let's move forward to the next question. The question said that in which one of the following preparation are you likely to come across cell junctions most frequently? In which of the preparation are you likely to come across cell junctions most frequently? Are they hyaline cartilage or the ciliated epithelium or the thrombocytes or the tendon? The answer will be in epithelial tissue. All types of cellular junctions are present. So ciliated epithelium is our answer. Okay, next, mark the odd one in each series. Odd one, we need to find the odd one. Okay, if you are talking about areolar tissue, blood and neuron and tendon, okay, which is odd one? Areolar tissue, loose connective tissue, blood, fluid connective tissue, neuron, neural tissue, tendon, dense connective tissue. So which is not connective tissue? Neuron is not a connective tissue. So this is the odd one. Next, RBC, WBC, platelets, cartilage. Cartilage is the odd one. All three are parts of blood. Now, exocrine gland, endocrine gland, salivary gland and ligament. So all are glandular tissue. Exocrine gland, endocrine and salivary gland. Ligament is a different tissue, that is fibrous tissue, dense fibrous tissue, dense regular connective tissue, right? So this is the odd one. Which of the following is not a connective tissue? Okay, which is not a connective tissue? So neuroglia is not a connective tissue, there is a neural tissue. So with this, I'm ending the chapter, structural organization in animals. Though it's not given in the new NCERT, but for our NTA syllabus and our upcoming NEET, we need to study the topic of animal tissue. So do not forget, and do not forget to check our PW app for all the one-shot lectures and the free materials like practice sheets and DPPs and handwritten notes of this particular class. And so bye bye for my side. Jai Hind, take care.